All right, lecture 20 is going to be on 3D analysis and analyzing protein structures. So the central dogma is that DNA encodes genes, so you transcribe DNA into RNA, and then RNA is translated by ribosomes into protein. And so these proteins each have their own unique functions. Example of that is hemoglobin, which transports oxygen through your blood. But there's all 25,000 genes, so there's 25,000 different proteins that kind of have their own unique function. And what determines the function of the protein is a combination of the structure, how exactly that protein is folded up, along with the chemistries of the amino acids. There's 20 different amino acids that can be used, and they all have different chemistries, different abilities to bind things. So if you want to know how a protein works, typically you need to know exactly where all of the amino acids of the protein are in three-dimensional space. And so this is possible. It is possible to determine this experimentally because of techniques that have been around for decades that allow you to first crystallize a protein and then solve its structure by sending X-rays through those protein crystals in order to create this X-ray pattern. So on the left, we can see the structure of hemoglobin. So the hemoglobin protein, which is composed of a tetramer of different subunits. So there's 4 different polypeptide chains that make up hemoglobin, and you're able to grow this into a crystal, which contains, billions to trillions of these protein molecules arranged in the exact same orientation. And so in the middle, you can see kind of some protein crystals, so you can grow them with different symmetries. They have different shapes, etc. But in the end, you can take these crystals and then pass X-ray through X-ray light that has a wavelength of X-rays, and then these X-rays will interact with the atoms of the proteins and deflect and create diffraction patterns. And so this is an example of a diffraction pattern that you might see. So the center is all of the X-rays that pass straight through without interacting with any of the atoms. So you typically blind that out. But then any of these dots, these dark colored dots, indicate where light is deflected with a very particular angle, and then you can use that in order to build this protein structure. So once you build this protein structure, you create a file, and this is called a PDB file. And the PDB file is stored in the PDB database. And this is the location of all the non-hydrogen atoms that are within the protein. So this technique is not able to identify hydrogen atoms. These are so small that they're not actually able to interact with the X-rays. And so you don't actually see these in the protein structures. So PDB files, they have their own format. It's one of the worst formats in the world. Hopefully you never have to write your own parser for that. But you can find PDB files by going to the PDB database, the protein data bank, and then searching for your protein of interest. So you can either search for the names of the proteins, or perhaps you can just search for the ID if you already know that. And so for this lecture and this first class, I'm going to give you the name of the protein we want to study, the name of the structure we want to study, and that is 3RIF. So that is a PDB ID that we are searching for. So we search that, and now we're able to find this particular structure. So this is the structure of a protein from C. elegans. It's a glutamate-gated chloride channel, and it's in complex with Fab, which are monoclonal antibodies, ivermectin, you might be familiar with this drug, and glutamate, which is actually the neurotransmitter that activates this channel. So this is a channel that's expressed in C. elegans neurons. It is a transmembrane protein, and so if glutamate is released by one of the neurons nearby, it goes into the synaptic cleft, and then this channel is able to actually sense the presence of that glutamate. It opens up, and then chloride is actually able to go through the channel from the extracellular environment to the intracellular environment, and then that changes the electropotential and leads to the neuron firing. Or in this case, chloride is a negatively charged ion. This actually prevents the neuron from firing. So this was identified, this structure was solved in 2011, and it was solved because ivermectin is a very important drug. It's a Nobel Prize winning drug that is used to kill, and it's an anthelmintic, it's used to kill nematodes, so parasitic nematodes. So this could be used in humans, so in many parts of the world there are still lots of parasitic nematode infections, so this is a very cheap, easy to produce drug that can be used to cure those, and it's often used in livestock and pets. So if you ever have given your dog heartworm medicine, most likely you're giving your dog ivermectin, and so if they get bit by a mosquito that's infected with the heartworms, the ivermectin will actually kill the heartworm before it's able to reproduce in your dog's heart. So we can use this structure to kind of study how exactly this protein works. So this is useful because we can see, the channel, so we can see what the physical structure of the channel looks like, and then we can see where the binding sites, where does glutamate bind in order to let chloride in, and then where does ivermectin bind in order to prevent that from happening. Ivermectin, of course, is now very popular as a potential COVID treatment. There are no real studies, I would say, that show that it actually that it actually improves COVID-19, but it is a very popular drug that people take these days. So you've probably heard of ivermectin in the news because of that. So why does it work to specifically kill parasitic nematodes? That is because humans and other mammals don't express glutamate-gated chloride channels. So we express other glutamate-gated channels like sodium channels, and we have other chloride channels that are that are activated by other neurotransmitters, but we don't actually use glutamate-gated chloride channels. And so this is a class of proteins that we simply don't express in humans and mammals, and so the ivermectin drug isn't able to actually bind and affect these proteins because we don't actually express them. But the nematodes do, and so the nematodes express these proteins, and so the ivermectin is able to bind them, activate the protein, and then eventually leads to paralysis and death for the nematode. So there are a couple things that this page will list. So one of the important things is the paper that published this. So it's always useful to potentially get more information by reading the paper, and often the paper will actually have more than one structure. Sometimes it'll only have one structure, but usually it has a lot of different structures. And so you probably want to know the structure of the protein in the presence or absence of certain ligands. So here we see there's a protein structure 3RHW, so this is also a glutamate-gated chloride channel in complex with Fab and ivermectin, so the glutamate is not present. And you can see some of the other different ligands that are used to study the protein structure. So you can see how the ligand influences the actual shape of the protein. So we're just looking at one particular shape this protein complex can actually have. Typically, there are multiple polypeptides that go into a structure. It's not just a single protein polypeptide. So for example, this has 5 different polypeptide chains. So this glutamate-gated chloride channel is actually a tetramer, and it's made of 5 individual subunits. And so we can get the names of those chains as A, B, C, D, and E. And if you wanted to know the gene that encodes this, GLC1 is the name of it, and you can click on that for more information. It's a 347 amino acid long. This is as I said before, this is a transmembrane protein, so it's typically very hard to create crystals out of transmembrane proteins because they're very hydrophobic. And so typically, and this is not just true of transmembrane proteins, but often you need to do some sort of protein engineering in order to form a crystal. This is why it's so hard and it takes so much time and so much expertise for structural biologists to actually solve structures. It might be your entire graduate career will be spent just studying the structure of a single protein, and you might spend 3 years actually identifying conditions where you can grow these protein crystals. And so in this case, they actually had to include the Fab monoclonal heavy chain and light chain. So there's also 5 polypeptides for that, F, G, H, I, and J, and then K, L, M, and O as well. And then small molecules, so you not only have polypeptides within these protein structures, but you also have smaller molecules. So these include ligands, for example, and ivermectin. So the name of ivermectin is IVM. So this is something that will be important to remember, the name of the molecule, so IVM. And so there's actually 1, 2, 3, 4, 5 different molecules within this crystal structure, and so that's within the D, the E, the A, and the B chains. And then there are some of these other molecules, so you can click on them to get more detail. These include NAG, LMT, UND, octane. So these are probably necessary for the structure of the protein. These could be like cholesterol type molecules. These are kind of fatty acid type molecules that somehow probably that bind to the crystal protein. Typically I ignore these proteins when I'm looking at that. But then the glutamate, so you can see that there's also the glutamic acid, so that's actually the neurotransmitter, so we can see where the neurotransmitter binds, and that's GLU. And then the chloride, there's also a chloride ion within that as well. So this, we can see where exactly the chloride travels through the channel in order to enter the cell. So whenever you are looking at a particular protein structure, it's good to go over the PDB and kind of understand what all is in this file that you're about to download. And even reading this paper, is probably a good thing to do. So you can I'm not currently on the VPN for Georgia Tech, but I could, for example, download the PubMed Central. And so you can look at the images, and this will also help you understand kind of what exactly you're about to look at, and read more information about it. So here we can see this is the transmembrane domain. So this is the part of the channel that goes into the transmembrane domain, and then we can also see, for example, this is the intracellular part of the cell, this is the extracellular part of the cell, and now they also show where the glutamate binds and where the ivermectin binds. And so this will help us find these molecules when we actually open this in the PDB. All right, so how do we visualize and look at these 3D structures? And so there's actually a large number of different software that you can download and use. If you work for a specific lab, you most likely, the lab will have their favorite version of that. So I use Ucsf Chimera, and that is what we are going to use for this class. So I'd like you all to download Ucsf Chimera. So you can find a link to that, and they've actually updated Chimera. So instead of using Chimera, which is the old version, they have this newer version called Chimera X. So we're going to use that. This is the first time I've actually used it as well. And so you can simply go to the download link, and I downloaded the latest production release. So if you want to use the exact same version as me, download the latest production release. If you have a Mac, you can download the DMG file. If you have Windows, there's this other releases tab, and so just make sure you're able to download it and get it running on your computer. So I have opened this, and I've previously opened this PDB file, but we are going to go back to here. So if you want to download that PDB file, you can simply download it, and then put it on your desktop if you would like. And then you can simply open that file, and now we can see it in our editor. You can also open it if you want in a text editor, if you want to see what this PDB file looks like. So it's a text file. It has all sorts of different metadata, and it is very difficult to parse because it requires specific number of spaces between different columns, so you can't like do tabs in between them. And if some of these things are longer than others, then you have to modify the number of spaces. So in general, you don't want to try to ever write a custom parser for this. But eventually you get to this section right here, and this lists the XYZ coordinates of all of the atoms that are within this file. And so we can see that right here. All right, so far we have been able to download, we've gone over a bit of the basics of PDB, and we've been able to download Chimera, and we've opened the glutamate gated chloride channel within that.